All right, Bismillah, Assalamu alaikum. So welcome to part 3 of chapter 7 lecture. Um, so as we talked about last time, this is the first slide from chapter 7 uh, lecture notes. And we're talking about uh, regulation of enzyme activities and how enzymes can be regulated, how their activities can be controlled. And we said last time that there are 4 major ways that cells use to control enzyme activity. And we have talked quite a bit about the first approach, which is called allosteric regulation. Um, and we talked about how enzymes can be regulated by something binding to a different site called the allosteric site. And what these compounds bind, these compounds are called effectors molecules or regulators, and they can be positive or negative. And it is basically because a conformational change occurs when things bind to them. So there's a conformational change that occurs when something binds and affecting its activity. Okay. Now, uh, I will not talk more about allosteric regulation. Again, we talked quite a bit about it, cooperativity and all those things that were all covered here. Today we'll talk about 2 different approaches called reversible covalent modification and proteolytic activation. So, in reversible covalent modification, the enzyme activity is controlled by something binding to it, a small group binding to it, and this small groups, small functional groups. An example really is phosphorylation. One of the most commonly studied approach is phosphorylation, where a chemical group, in this case, we have a chemical group such as phosphate is covalently added or removed from the protein, and this modification can either activate or turn off the enzyme. And again, as we said, the most common uh, modification is called phosphorylation, um, where a phosphate group is added to the protein. So you have a protein, for example, like this, right? Um, and something happens to it and it changes its conformation and uh, it becomes thing a different conformation, and this is because a phosphate group is added to it, right? And the addition of a phosphate group is an example of covalent modification. Now these phosphate groups have to be added somewhere, and they're added to the amino acids, these 3 amino acids, um, because these 3 amino acids have OH group. So phosphorylation is a modification which is added to the OH groups of amino acids, and there are 3 amino acids that have OH groups: serine, threonine, and tyrosine. And you add a phosphate to this OH group. And phosphate, of course, is coming from somewhere, and it has to come from ATP. Um, the phosphate ATP stands for adenosine triphosphate. It gives a phosphate to the protein and becomes ADP, right? And this is what happens. So this is called example of phosphorylation. Now again, in this case, you could have an inactive protein which will become active by phosphorylation, right? Um, so, but in some cases, again, it doesn't have to be. In some cases, it could be, you know, turn off the activity as well. So it's possible that you can have an inactive enzyme which becomes active, or you can have an active enzyme. When you add phosphate to it, it becomes inactive. Okay. So here's an example. You have an enzyme called pyruvate dehydrogenase. This enzyme is active in the unmodified. So there is an OH here which is not phosphorylated. So it is active in the unmodified state, and ATP comes along. It gives a phosphate to the hydroxy group. It becomes ADP, and then you have this phosphate added here on the protein. Now in this case, they're showing that the enzyme becomes inactive, right? So again, phosphorylation can cause things to become active, or in some cases inactive. So don't always think that phosphorylation causes things to become active. Okay. Now this process of transfer of phosphate from ATP to OH, is catalyzed by enzyme, and the enzymes are called kinases. So there are enzymes called kinases, and kinases, they add the phosphate group, they phosphorylate uh, proteins. Now we said these are reversible modification, reversible covalent modification. So the modification which is adding a phosphate is reversible, and it can be reversed. You add water and you can release a phosphate, inorganic phosphate. And this reversal, so if it was inactive, now it becomes active. This is done by an enzyme, and the enzyme is called phosphatase. So the enzyme called phosphatase is our enzyme that removes the phosphate group, right? So again, it's a very simple mechanism. You have a protein which is active, and then if you want to make it inactive, you add a phosphate group to it, it becomes inactive. Some other enzymes or proteins would be inactive to start off with, and then you add the phosphate group, it becomes active. Now this, if you don't want it to be active, you can reverse it. You can add water and phosphate comes back, and so you can recycle between active and inactive states, and this happens very, very quickly and is a very efficient way of controlling enzyme activity. Okay. Now, again, as I said, the phosphorylation that we talked about is added to the side chain OH groups of amino acids, and there are three amino acids: serine, threonine, and tyrosine. You do not have to know the structures of all three of them, but you should know the names. But you should know the structure of course, serine, and serine has OH group. The phosphate reacts with it, and then you add a phosphate, and you have a ADP here. But the same process happens for tyrosine, where you have this, or threonine, where you have OH group here as well, right? So phosphorylation by ATP can convert inactive enzyme into an active one, or reverse it, right? All opposite, or go in the opposite direction, or reverse. So please make sure that it doesn't mean that it's always inactive to active. That is not always the case. In some cases, as we saw in the last slide, you can have an active enzyme which is not phosphorylated, and if you add the phosphate group, it becomes inactive. Now, membrane transport is actually another very nice example of uh, controlling activity of a transporter by phosphorylation. So not only enzymes can be regulated up and yes and no with phosphorylation, or active and inactive with phosphorylation, but other proteins like transporters, like membrane transporter, can also be regulated by phosphorylation, and we'll see this in the next slide, right? Um, so in this case, again, um, uh, some of you may know that potassium and sodium are ions that cannot cross the membrane. So there's a lipid membrane. So this would be the membrane. So this is a lipid membrane, for example, right? And if we need to move potassium out or sodium inside, you cannot go through the membranes, because membranes are lipophilic. They have hydrophobic. These are charged ions. They cannot go through the membrane, and so there has to be a transporter. So here's an example of a transporter, and we call them sodium potassium pump, and I think we'll see it in the next slide. So in this case, you have a transporter um, which is non-phosphorylated, right? And sodium can bind to it, and binds to it. Um, it becomes uh, it gets phosphorylated, and phosphorylation will cause the potassium to go from outside to inside, and then the phosphorylated form of the receptor can bind the sodium, and then when it comes inside, it becomes unphosphorylated. So this is shown a little bit better in the next slide, where the role of phosphorylation is more clearly explained. So when ATP is hydrolyzed, energy is released, and you phosphorylate the receptor, which causes a conformational change, which causes the ions to move back and forth. So let's look at this another example. So in this example, they're showing you the lipid membrane very nicely. Right, this is guy here. Okay, and what they're showing you that you may have you have a lot of sodium on the outside, and you have a little bit on the inside, and then you have a lot of potassium on the inside, and you have a little bit on the outside, right? So inside inside you have 100 millimolar potassium, and then outside you have 150 millimolar sodium. So if you need to push sodium from inside to outside, right? How can you do that? You have so little here and you have so much outside, so it's not going to go, it doesn't like to go against a what's called a gradient, a concentration gradient. We know things can go down a gradient, so 150, if you have 150 outside and 10, it can go inside from 150 to 10 easily. But to go from 10 to 150 in the opposite direction is not easy because again you're going against concentration gradient. So here's an example. So you have the transporter, sodium potassium pump. That's the name of the transporter, sodium potassium pump. And normally, on the inside, as I said, you have a little bit of sodium. So the sodium from the inside of the cell, of the membrane, will bind to the receptor. And when it binds to it, nothing happens. It's just bound like this. And there is an opening which doesn't allow the sodium to go out. So you just bind to it. But then ATP comes along and ATP phosphorylates the receptor. So once you have a phosphorylation here, there's a conformational change. See, if you look at this shape and you look at this shape, they're very different. So the conformational change such that this mouth opens up and allows the sodium to go out now. So shifting sodium from inside to outside occurs because of the phosphorylation of the receptor. Okay, so once you have pushed it out, what happens, potassium again is on the outside, and you don't have too much. You have 5 millimolar potassium, so it binds to it. And when binding of potassium, it binds inside, but binding it doesn't allow it to come out. It's not going out because it is phosphorylated. So once you remove the phosphate from the receptor by adding water, so when this is gone, there is a conformational change, and then at the receptor, the transporter opens up, and now you have moved potassium from outside to inside, right? So that is example of a sodium potassium pump using phosphorylation to actually move sodium ion from inside to outside, Um, and again, please note this is against concentration gradient. It's going from low to high. That's the key point. And then it allows potassium from outside to come inside, but also against a concentration gradient as shown here. Okay, so that's another example of how phosphorylation can be used to control the activity of an important protein. In this case, this is a sodium potassium transporter or sodium potassium pump. Okay, so the third way of controlling enzyme activity, so we've talked about allosteric regulation on the first way, we've talked about reversible covalent modification second, and third one we'll talk about what's called uh, proteolytic cleavage of proteins, or conversion of or cutting off a protein, clipping off part of the protein. So in this way of controlling enzyme activity, you have enzymes that is made in an inactive form, and since they're not active, we call them proenzyme, like before they're ready to be enzyme, proenzymes. So proenzymes are enzymes that are made in inactive form, and these proenzymes can be converted to the active form by proteolysis. So you have an enzyme. So for example, this is an enzyme like this, and what you do, you somehow chop off this part. You break off this part. Part of the uh, part of the chain is broken off. Once you've done that, then this becomes active. So you hydrolyze part of the enzyme. This guy is gone, and then this thing will become active, right? And that is called regulation of enzyme activity by proteolysis. And this happens basically when it is needed inside the cell, right? So it's made in the inactive form, it's circulating all over the place, and when you need it to be active, you cut off part of the protein, and then it becomes active. And the example that we will talk about are these digestive enzymes. We talked about this in the class a couple of weeks ago. I hope you remember that. So there's an enzyme in our stomach called pepsin, Pepsin is the active form of the enzyme, but it does not is not made in the active form. It is made as pepsinogen. Pepsinogen is synthesized and it is inactive. In the stomach, when it goes there, then it's cleaved. You cut off part of pepsinogen and becomes pepsin. Another example is trypsinogen, which is again this part has to be chopped off, and when you remove that part, it becomes trypsin, which is active, right? And there are many other enzymes that actually, and we talk about fibrinogen and fibrin as well, same idea. So these proenzymes are also called zymogen. So zymogen is also same as proenzyme, right? It's exact same thing. So these are inactive precursors, right? They're not, and they're when if you cleave off part of them, then they become active, right? So pepsinogen, trypsinogen, and so on and so forth. And chymotrypsinogen is another example, and we'll study quite a bit about chymotrypsinogen to see how it works and how it can be regulated by proteolysis. So it's synthesized, this enzyme is synthesized in the pancreas, and is a single polypeptide chain, right, of 245 amino acids, and has 5 disulfide bonds, right? But again, if it's synthesized, it would be inactive. Now it's secreted in the small intestine. In the small intestines, there are proteolytic digestion. It chops it off and makes it into an active form. So this is shown here a bit better. So this is the zymogen, or the inactive form, or the proenzyme. This is called chymotrypsinogen, right? This is what this guy is made, and it is inactive. It has 5 disulfide bonds, 1, 2, 3, 4, 5, right? And it has 245 amino acids total. Okay. So there's an enzyme called trypsin. So trypsin, or it could be trypsin, it could be something else, but trypsin comes along and it cuts here, right? One cut. And again, nothing happened. Then there's, but what it does, it actually makes it into a little bit active form, so it starts cutting itself. It chops this part out and takes this part out. So this is removed and this is removed. So there are 2 amino acids here and 2 amino acids here. So you have lost 4 amino acids now, right? So you remove 4 amino acids, and that what that leads to, it leads to this alpha chymotrypsin that's shown here, right? Which again, if you look at this amino acid number 1, it goes to 13, and then 14 and 15 are missing, and it jumps to 16, right? So now you can see there's a change in the primary structure. So from here to there, right, there's a change in the primary structure because 2 amino acids are missing, right? And then these 2 are missing as well, so you're losing 4 amino acids totally. So change in the primary structure from this to this also causes a change in the tertiary structure, and this guy is inactive, and the alpha chymotrypsin is active, right? And again, this inactive to active occurred because you're removing some parts of the protein. Okay. Um, Now, again, don't read all of this thing. I mean, don't memorize all of this thing. And this is really the most important part, right? The most important part is shown here, that during the whole process, there is change in the primary structure, and the primary structure change results in changes in the secondary and tertiary structure of the protein, and that's what makes it an active protein. Okay. But don't worry about all the details, and there's no need to memorize. Just read that, but don't memorize these details. Okay. Now, well, I'm going to stop here, and we're going to talk about in the fourth lecture series. We'll talk about what is meant by the active side of enzymes. We'll talk now a lot more about active side and catalysis and our enzyme activity, how enzymes do what they do, by looking at it a bit more, inshallah. All right, stop that. Thank you.